For this question, I will go over the histology of the lymph node and explain the type of immune cells that is present in each region of the lymph node. So if you notice here, there are multiple afferent lymphatic vessels that is going into each lymph node. So there are multiple afferent lymphatic vessels, versus there is usually one or two efferent lymphatic vessels that is coming out. Then we have the capsule, which is the outer region. Then we have the cortex, and then the medulla is this red region that is inside at the center. The medulla is connected to the efferent lymphatic vessels. Now the cortex contains follicles, which are all these purple structures, and there are two types of follicles. One is primary follicles, which is all the same color all along, so we don't have a pale center, and these are dormant. Versus the secondary follicle, which has a germinal center here. So these are actually active follicles, which means that B cells that are located in follicles have undergone maturation, and they do that by developing hypermutation and also undergoing immunoglobulin switching. So just to repeat that, we have these primary follicles which are inactive versus secondary follicles which have germinal center there, in which B cells are undergoing maturation. Then the region between medulla and the follicles, which is this blue region shown here, is referred to as paracortex, and it contains T cells. Now, since this region contains T cells, any condition that increases the number of T cells will cause into the enlargement of this region. For instance, if someone develops a viral disease, and thus there would be an extensive immune response against the viral disease, the T cells will increase in this region, and therefore this region will expand. On the other hand, if someone has the DiGeorge disease, in which there is no T cells, as a consequence of which the paracortex region of the lymph node will be underdeveloped. Then we have the medullary cords, and medullary cords are dense structures that are inside the medulla of the lymph nodes, and it contains the plasma cells. And finally, we have the medullary sinuses, and medullary sinuses is the space in between the medullary cords, and it contains the macrophages, which functions in the filtration of the lymphatic fluid. And finally, we will have the reticular cells. And what the reticular cells do is that they make reticular fibers, and so by forming reticular fibers, they direct B and T lymphocytes into different regions of the lymph nodes. Now that we've covered this, let's go over the actual histology of a lymph node and repeat everything that I just explained there. So we have two types of follicles: primary follicles like this, which are dormant and have not undergone maturation, versus secondary follicles where the B cells have undergone maturation and thus form the germinal center right here. So this is called the germinal center. And then the region between the medulla and the follicles is called paracortex, and it contains T lymphocytes. So if someone has a viral disease, the paracortex will be enlarged, versus if someone has the DiGeorge disease, which has no T cells, then this region will be underdeveloped. Finally, we have the medullary cords, which are these dark structures that are shown here. There is another one here. You can see another one right here, and one more here. Like, there's plenty of them out there, and so they contain plasma cells. And finally, we have medullary sinuses. So it's these pale regions that is in between the medullary cords, and it contains the lymphatic fluid and is connected to the efferent lymphatic vessels. So the medullary sinuses contain two types of cells: macrophages, which will filter the lymphatic fluid, and reticular cells, which will make reticular fibers to direct B and T lymphocytes into different regions of the lymph nodes. Now, if you look at the type of cells that is present in a lymph node, you can already tell what is the function of a lymph node. It is used for storage and activation of B and T lymphocytes. It is used for immunoglobulin production, and it's also used for filtering the lymphatic fluid by the macrophages.